Welcome to our channel, Mootech. Today we are beginning a fascinating journey into the tools that let us see the unseen. This presentation, Microscopy in Microbiology, will explore how we uncover the invisible world around us. Now, let's discuss why microscopy is so crucial. This introduction covers the fundamental need and importance of these instruments. Microscopy reveals the invisible world of microorganisms undetectable by the naked eye. Just imagine, entire universes of life exist completely hidden from our direct sight. It enabled pivotal discoveries ever since Antony van Leeuwenhoek first observed animalcules with his handmade lenses, sparking a scientific revolution. Today, it is essential for modern clinical diagnostics, pathology, and medical research, forming the backbone of disease investigation. Critically, it links specific microbes to disease causation, known as etiology, and directly guides treatment decisions. Ultimately, it facilitates advancements in therapy, drug development, and public health policies that protect communities worldwide. Moving on to the core concepts. This slide explains the fundamental principles that make microscopy work: magnification and resolution. Magnification, abbreviated as M, is the apparent increase in size of an object compared to its actual size. Think of it like zooming in on a picture on your phone. It gets bigger. Resolution, often noted as D, is the minimum distance between 2 points that can still be distinguished as separate. This is about clarity. You can zoom in all you want, but if the image is blurry, you can't see the details. There is a crucial trade-off here. High magnification without good resolution results in empty magnification, a big but useless blurry image. The numerical aperture, or NA, of the lens determines its light gathering ability and resolving power. A higher NA means a better ability to distinguish fine details. Also, using a shorter wavelength of light leads to better resolution, meaning a smaller d. This principle is why electron microscopes, which use very short electron waves, are so powerful. Now, let's categorize our tools. This slide provides a classification overview showing the different families of microscopes based on their light source and how they work. We have the basic simple and compound optical microscopes that use visible light. Then we have specialized types that enhance contrast, like dark field and phase contrast, perfect for seeing live transparent cells. Fluorescence microscopy uses glowing molecules to tag specific parts of a cell. And for the ultimate detail, we have electron microscopes like TEM and SEM, which use beams of electrons instead of light to see at the nanoscale. Let's start with the simplest type. In this slide, we look at the simple microscope. It consists of a single convex lens or a magnifying glass system. It's the most basic tool for magnification. Its magnification range is typically limited to 10x to 25x, so it's great for looking at insects or small parts, but not for bacteria. It's often used for field checks, quick screening, and dissecting small organisms, a handy tool for biologists on the go. Its advantages are clear. It's extremely portable, low cost, and easy to operate. However, its limitations include low resolution and susceptibility to image distortions called spherical aberrations, which is why we need more complex scopes for microbiology. Next up is the workhorse of the lab. This slide covers the compound microscope. It uses a two-lens system, an objective lens and an eyepiece lens working in series. The total magnification is the product of both the lenses. For example, 40x from the objective and 10x from the eyepiece gives you 400x magnification. Key parts include the condenser to focus light, the diaphragm to control it, the stage to hold your slide, and focus knobs. It's the standard tool in teaching and diagnostic labs. A key technique is using the oil immersion objective, usually 100x. Placing a special oil between the lens and the slide minimizes light scattering, allowing for the highest resolution images possible with light. Now, let's explore a technique that makes transparent things shine. This slide introduces dark field microscopy. Here, the central light is blocked. Only light scattered by the specimen enters the lens. This makes the specimen appear bright against a dark background, like stars in the night sky. It's excellent for observing thin, live spirochetes like Treponema pallidum, the cause of syphilis. A major benefit is no staining is needed, so you can watch live bacteria swim. It also highlights edges and fine structures with fantastic contrast. Continuing with techniques for live cells, this slide explains phase contrast microscopy. It works by converting invisible differences in cell density and thickness into visible differences in brightness and darkness. The key components that make this happen are an annular diaphragm in the condenser and a phase plate in the objective lens. This makes it ideal for observing unstained living cells like bacteria, sperm, or tissue cultures. You can watch live processes happen in real time. It visualizes details like organelles and cell division without harming the cells with chemicals. Now, stepping into more advanced methods, this slide covers fluorescence microscopy. It relies on fluorophores, molecules that absorb high-energy light and then emit lower-energy light of a different color. The microscope uses special filters and mirrors to shine exciting light like UV or blue and then isolate only the emitted glow. Common stains include DAPI that makes DNA glow blue and FITC for green fluorescence. Techniques like immunofluorescence use antibodies to tag specific proteins and FISH uses probes to highlight genes. This allows for rapid pathogen detection and pinpointing exactly where a protein is inside a cell.
Now, let's push beyond the limits of light. This slide asks, electron microscopy, why and when? The answer is in the wavelength. Electron microscopes use beams of electrons, which have a much shorter wavelength than visible light. This allows them to achieve ultra-high resolution, down to the sub-nanometer level. We can see viruses and molecules. It requires a high vacuum and complex sample prep. The two main types are TEM for seeing inside and SEM for seeing surfaces. It's crucial for visualizing viruses, organelles, and the fine architecture of cells. Diving deeper into electron microscopy, this slide focuses on the transmission electron microscope, or TEM. Its job is to visualize the internal structure of cells at an incredible resolution. How does it work? Electrons are shot through an incredibly thin specimen. Electromagnetic lenses focus the beam and the image is formed on a screen, much like a slide projector but with electrons. Its capabilities are staggering, with magnifications from 10,000x to over 2 millionx. Preparing a sample is intense. It must be sliced ultra-thin, stained with heavy metals and placed in a vacuum. Its applications are vital in cell biology, for seeing organelles and is especially important for identifying the detailed structure of viruses. For stunning 3D surface views, we use the scanning electron microscope, or SEM. This slide explains its function. The SEM scans a focused electron beam back and forth across a specimen's surface. Detectors pick up the secondary electrons kicked off the surface to create an image. This gives a spectacular 3D-like view of surface topography with great depth of field. Samples, especially biological ones, must be carefully prepared, fixed, dried, and coated with a thin layer of metal to conduct electricity. It's used to study the surface texture of spores, pollen, biofilms, and also invaluable in material science. Now let's break down how a basic light microscope works. This slide walks through the mechanism step by step. First, the illumination source provides light. Second, the condenser lens focuses that light into a cone onto your specimen. Third, light interacts with the specimen. Some passes through, some is absorbed. Fourth, the objective lens captures this light and creates a magnified real image. Fifth, the eyepiece lens magnifies that image again for your eye. A special technique, oil immersion, is used with the strongest objective to reduce light loss and get the clearest possible picture by matching the refractive index. Similarly, let's see how an electron microscope works. This slide outlines its principle. First, an electron gun generates a beam of electrons. Second, electromagnetic lenses focus this beam. Third, the beam interacts with the specimen, either passing through it in a TEM or scanning its surface in an SEM. Fourth, detectors collect the resulting electrons. Fifth, these signals are processed to form an image on a screen. A critical requirement is a high vacuum throughout the entire column to prevent air molecules from scattering the electron beam. Now, where do we use all these tools? This slide covers the practical applications of microscopy across many fields. In clinical diagnostics, it's used for analyzing blood smears and tissue biopsies. For microbe identification, it's all about observing cell shape and staining results like the gram stain. In research and development, it helps study cell structures and test new drugs. In quality control, it ensures products are sterile and safe in pharmaceuticals and food. In environmental science, it monitors water quality and studies soil microbes and biofilms. So, why is microscopy so powerful? This slide lists its key advantages. The first is direct visualization. You get immediate visual proof of what's there, its shape, and how it's arranged. It offers incredibly high magnification, especially electron microscopes that can see viruses. There are multiple modalities, different techniques for different samples, from dead-stained ones to living cultures. Many methods are non-destructive, letting you watch live cells move and grow. Finally, targeted labeling with fluorescence allows you to hunt for specific molecules with high precision. However, there are challenges. This slide discusses the limitations of microscopy. Sample preparation can introduce artifacts, unnatural distortions that mislead the observer. The high cost of advanced instruments like electron microscopes limits access. Light microscopy has a fundamental resolution limit due to the physics of light, so viruses remain invisible under standard scopes. Operating these tools requires significant skill and training. Finally, interpretation can be subjective, requiring expertise to tell real structures from debris or artifacts. Finally, we've reached the end of our visual journey. Thank you for watching our presentation on microscopy. Don't forget to subscribe for more biology content from Mootechie. Stay curious and keep exploring the microscopic world. We hope this look through the lens has inspired you to appreciate the incredible tools and techniques that reveal the hidden details of life. Keep questioning, keep discovering.